get rid of that. Now let's bring up that. All right. Introduction. This is very introductory. We're going to go over just some basic components, organization of the nervous system. Before I get started, I'm a big guy on simplifying things. Let me just pass some of these out. This is a little like just a very basic overview of the nervous system and how the different parts relate to each other, and just so you can see it all on one page. And that's kind of what we use as some of our guide today. So let's see if we got enough here. Might have to share some with another table if I don't give you enough here. There you go. Oh, there's 5 tables here. Here, take some of that. That. Make sure you guys share with each other. Pass it to another table. I think I brought like 42 copies. There's 40 of you guys? 39? Okay. Should be a few extra copies out there. All right, we're starting basic. Neurons. So basic components of the nervous system are neurons. Then there are support cells that help support the neurons, the neuroglial cells, the glia, glue that holds the neurons together. We have a central nervous system, right? That's our brain and spinal cord primarily. We have a peripheral nervous system, everything outside the brain and spinal cord. And we have an autonomic nervous system, which is contained both within the peripheral and central nervous systems. It's just kind of one of the things they pull out to talk about different parts of the nervous system. So 4 main basic components. So this is your little handout I gave you that kind of shows. So you have the nervous system on the left. We just mentioned these 4 basic components. And as you go to the right, we just start splitting off into the more subcomponents of each of those particular structures, okay, or parts of the nervous system. Okay, so just, we're not going to go through this step by step, but just as I'm talking and going through the slides, it's sometimes hard to understand where are we at in this thing. This is your kind of reference guide to go back to, okay? Starting with neurons is what we're going to start with first. Basic structural functional unit of the nervous system. This is how the nervous system works, or by communication between neurons. It's how you convey information. If a neuron brings information towards the central nervous system, or that neuron brings information towards a structure within the nervous system, it's called an afferent neuron. I think of the A in afferent like acquire. It's acquiring information or gathering information. So it's coming towards that structure or towards the central nervous system, an afferent neuron. Okay? On the opposite side of that, there's efferent neurons. Efferent neurons take information away from the central nervous system towards the periphery, like if you're sending a signal to a muscle to make it contract, that would be an efferent neuron. Or if you're sending a signal from one region of your brain to another structure, that neuron taking it away from one structure towards another structure could be also an efferent type neuron. Just so be aware of that. Mainly we're talking about communication between central and peripheral nervous system with these afferent and efferent neurons. Okay? However, there's some of the same arrangement within internal structures when you're communicating between structures within the nervous system. Neurons that connect only with other neurons are called interneurons. So there's interneurons. And actually, interneurons make up about 90% of your neurons. So the big, long neurons that communicate between structures, between your peripheral and central nervous system, those are the ones we talk about the most. But you have all these interneurons that also convey information and modify input and output. And so they really kind of help kind of fine-tune the nervous system. And they're 90% of your nervous system, okay, is the interneurons. Get my little clicker here. All right, that's the neurons. Then we talk about the glial cells, the neuroglial cells, the neuroglia. The neuroglia is really how you pronounce that. Sometimes just referred to as glial cells. These are non-neuronal cells, so these don't generate action potentials. Neurons generate action potentials and can actually send and propagate this thing down the neuron. Neuroglial cells don't do that. They're there to support the neurons, okay? Now with new research, they're showing that neuroglial cells can influence the function of the nervous system, and even there are some types of pseudo kind of signals sent via electrochemical gradients and things like that, but it's not truly an action potential. But anyway, don't worry about that right now. They insulate. They nourish, they support. So if you look at the types of neuroglial cells, which I updated this one slide before you guys got your copy, so if you're looking for it, it's not on there, at least the picture there. I just found that real quick because I thought it was nice to see. If you look at the types of neuroglial cells, you have some in the central nervous system and some in the peripheral nervous system. They're a little different between those two. The ependymal cells that you're seeing up there, those line the ventricles where your cerebral spinal fluid is. There's like a little layer of cells there that line that. The oligodendrocytes in the central nervous system, they form the insulation around the axons that are in the central nervous system, your brain and spinal cord, that allows your signals to be sent faster. The astrocytes have those little end feet on them. They are the glue. They're kind of just in there, packed in there around all the neurons to provide kind of substance. And those little end feet also wrap around blood vessels to form these little thing called tight junctions to prevent cause the blood-brain barrier. So viruses and bacteria have a harder time getting into the brain and central nervous system. The microglial cells there, those are the little guys that have phagocytosis that go around, eat up leftover dead cells, try to kill viruses and bacteria in the central nervous system and the microglia. In the peripheral nervous system, don't worry about satellite cells. Just Schwann cells are the cells that also wrap around the axons in the peripheral nervous system to create insulation. Those are the equivalent of an oligodendrocyte in the central nervous system. So those two provide the same function. The oligodendrocytes in the central nervous system, Schwann cells are in the peripheral nervous system. A disorder like multiple sclerosis, it attacks the oligodendrocytes in the central nervous system and causes damage to the Schwann cells. Or something like a disease like Guillain-Barré syndrome, it attacks Schwann cells in the peripheral nervous system. They have a similar type of mechanism. It's an autoimmune type response that damages it, but MS is in the central nervous system. Guillain-Barré is in the, and you don't have to know all that. I'm just giving you guys some clinical examples so you know why you need to know this eventually. So now we went neurons and neuroglial cells. Now we're going central nervous system, okay? As we're going to talk about that. Generally considered to have 7 main parts. Some textbooks will not have 7 parts, but anyway, we're going to go this route with 7 main parts. You have the cerebral hemispheres, okay? The thing you think of when you think of the brain proper, just these big, wrinkly thing up here in your brain. That's the cerebral hemispheres. Deep inside those cerebral hemispheres there's a couple structures called the diencephalon. Two, mainly made up of the thalamus and the hypothalamus, okay, which is deep down in the cerebral hemispheres. Then we have the brainstem, which includes the midbrain, the pons, and the medulla, which are each considered different parts of the nervous system. So midbrain, pons, medulla. Then we get to the cerebellum, this big other wrinkly thing, kind of the hindbrain or the area of the brain that's sitting off the back and bottom of your cerebral hemispheres. And then the spinal cord, okay? Those are the 7 main parts. So there they are. You can kind of look at the cerebral hemispheres on the top. The diencephalon is the pink-looking area, almost kind of looks like a little egg in the middle of the brain there, and you guys will get to see this on the specimens here. And then the midbrain, which is, if you look at the midbrain there, that brown, the darker brown structure, or what, is that brown? I don't know. I'm colorblind. But it's right below the thalamus and hypothalamus, the diencephalon, but it's really kind of almost up in the middle of the brain. That brainstem really sticks up inside of there. So the brainstem includes that midbrain, and then the pons, that little kind of more bulbous structure there below the midbrain, and then the medulla. Sitting off the back is the cerebellum, and then the red, once you pass through the foramen magnum, see the hole in the bottom of the skull, the foramen magnum, that's the division between your brainstem and now spinal cord, okay? Peripheral nervous system. Peripheral nervous system now, so we covered central nervous system, now we're moving peripheral. All parts of the nervous system that are not encased in the skull or vertebral column, okay? So basically your cranial nerves and your spinal nerves, okay? So everything coming out of your brain and brainstem and your spinal cord that is not encased in that, okay, are considered peripheral nervous system. So there, a picture there. Everything in that gray, that lighter grayish blue is central nervous system, but all the yellow is peripheral nervous system. So just an appreciation for what's going on there between those 2 major components. Autonomic nervous system is the next main component here, okay? So components in both the central nervous system and the peripheral nervous system. So the autonomic nervous system has parts that are important to it that lie within the central nervous system, like the hypothalamus is the master regulator of your autonomic nervous system. That's in your brain, so it's part of the central nervous system. But you have to have nerves that go out to affect your organs, your skin, your blood vessels. Those are peripheral nervous system. You have to send those autonomic signals to those to do that. We have a sympathetic part of this and a parasympathetic part of the autonomic nervous system. The sympathetic, you guys have probably heard of, kind of typically the fight or flight, kind of ramps up metabolism, makes your heart rate go up, makes respiratory rate go up, those kind of things, blood pressure increase. That's the sympathetic nervous system, okay? Often also called the thoracolumbar division of the autonomic nervous system. So if you hear thoracolumbar division of the autonomic nervous system, that's analogous, or it's synonymous with sympathetic nervous system. It's just because the nerves that take that information, the sympathetic nervous system information, to go to all your organs and your skin and all that, they come out between the thoracolumbar region. They exit between T1 and L2 is where all your sympathetic outflow comes out of between that thoracolumbar region. That's why it's called the thoracolumbar section or division. The parasympathetic, also known as kind of the need and feed for storage of energy and those kind of things, is also referred to as the craniosacral division of the autonomic nervous system, okay? Because it has cranial nerves that go down to influence your organ systems and so forth, and a few sacral nerves that also do the same thing, especially down to your lower colon and your bladder and your genitals and so forth, the parasympathetic control. So it's not coming out in that thoracolumbar region. That's all sympathetic. The parasympathetic, it's around the sympathetic. It's craniosacral. It's cranial nerves, a few important cranial nerves that do that, and then sacral nerves that supply the parasympathetic control. Both the sympathetic and the parasympathetic tend to innervate smooth muscle, okay, not your striated muscle, but more smooth muscle, cardiac muscle, and secretory glands. They tend to have opposing actions, not always, but sympathetic and parasympathetic tend to have opposing actions. Sometimes they have complementary actions, especially like in sexual function and those kind of things. They have some complementary actions. But they mainly are geared around maintaining homeostasis, whatever you need to maintain normal homeostasis under different environmental conditions and under different stresses, okay? So that's just really quick on autonomic nervous system. So if you look here, you'll see on the left a sympathetic nervous system, and you'll see where all of the sympathetic nerves that are going to the different organs and sweat glands and skin and all that kind of stuff, they all end up coming out of that T1 to L2 region, okay? That's the thoracolumbar division, okay? Then on the right side, we have the parasympathetic division, which is going to be coming out of the cranial nerves. So you see the cranial nerves up top coming out from the brainstem there, going to the various regions. And then down lower, you have a few sacral nerves that go down to supply the parasympathetic control of the organs of the pelvis, okay? So that's just kind of an appreciation for that. In the sympathetic nervous system, in parasympathetic, there's two neurons in that pathway. In the sympathetic nervous system, you have a short, we call preganglionic neuron. So before it makes a connection within a ganglion to the longer postganglionic neurons. So you can see there's kind of a shorter blue neuron, then there's a longer like red neurons, and there's also some blue ones down there too. But just appreciate there's like a shorter preganglionic, then a longer postganglionic. And it's the opposite for the parasympathetic. You tend to have a long preganglionic neuron, then it makes a synapse closer to the actual effector organ it's going to be affecting, and a little short postganglionic neuron. All right, that was like super quick, brief overview of basic components of the nervous system. Okay, neurons and cerebral hemispheres and all that kind of stuff, and peripheral, central, autonomic nervous system. Now we're going to get into some language, because if you know language, it's just easier to interpret things when you're reading and those kind of things. So that's what we're going to talk about here. Is there's certain terms that I was like, that's all they're talking about. That's not something super complicated. That's just what you're going to be able to appreciate with this. So first of all, let's take a look at orientation and some of the terms used to describe where parts of the nervous system are, relationship to each other, and so forth. So it's important to understand that the nervous system, when we're talking about, I'm going to draw on my screen here a little bit. It helps for doing this. So when we talk about, I'm just kind of outlining the brain stem, which extends up into the brain there, and the spinal cord. You'll notice that the brain stem and the spinal cord tend to be in normal alignment with the body's position, okay? And so when you look at that, if you look at that brainstem and spinal cord, it aligns with the anatomical axis of the body, right? It's kind of lined up with it. When you get up into the cerebrum and the cerebellum, there's a little kink. This is called the neural axis. So the neural axis in the brain and spinal cord aligns with the body's axis, anatomical arrangement or axis. But once you get up into the cerebrum kind of goes boop, and it kind of tips over like this a little bit at a slight angle. And so that neuraxis is slightly oblique in the cerebrum and the cerebellum, okay? So the main point you have to understand that is when you're reading about like locations, if somebody's describing where a certain part of the brain is in relationship to another part of the brain, they use terminology to describe that relationship. Is it superior? Is it dorsal? Is it anterior? Is it posterior? Is it inferior? Is it rostral? Is it caudal? Those kind of things. So if you just have this picture in your head as you read that, you can make sense of terminology. So it's easy in the brainstem and spinal cord because it aligns with what we typically think of as the body's normal axis. So, in this case, if you say something in the spinal cord is dorsal or posterior, so this is dorsal and posterior, those are synonymous terms when you're talking about something in the spinal cord or the brainstem, okay? So we have in the spinal cord, we have the dorsal horns. You could also call them the posterior horns, okay? This is gray matter. And so dorsal and posterior are synonymous with each other in the spinal cord and in the brainstem, okay? Caudal and inferior these are synonymous with each other in the brainstem and spinal cord, okay? Caudal's towards the tail, and inferior, okay? Ventral, I know what I need to do, hang on. I had this last time, I had a problem with this. Animations, no, not animations, transitions. Do not advance slide on mouse click, there we go. So ventral here in the spinal cord is synonymous with anterior, okay? So this is anterior, so that's synonymous with anterior. And then superior with this is, if you took this little arrow right here and you made it and you lined it and just went straight up like this would be the same as rostral. So superior and rostral are synonymous when we're talking about spinal cord and brainstem. That's all we're talking about right now, okay? So just showing you that when you sort of describe like a certain, part of that nervous system, so something is rostral or superior. So the C7 nerve root is rostral or superior to the T1 nerve root, okay? You could use either one of those terms and that would be okay to call it that way. But now when we get into the cerebrum, if we're talking about and you're reading about some structures in the cerebrum, in this case, since now the neuraxis tilts, it's no longer going straight up this way, now what happens is these terms are a little different. So now in this case, caudal becomes synonymous with posterior. Where down in the spinal cord and brainstem, dorsal was synonymous with posterior. Does that make sense? So you just got to understand, once you're up in here, if somebody's referring to a structure, like the occipital lobe is now caudal or posterior to the, parietal and temporal lobe. So you could use either one of those terms. You could say it's caudal or posterior. And then when we get up to the dorsal, so in the cerebrum, dorsal is synonymous with superior. Where in the brainstem and spinal cord, dorsal was synonymous with posterior. I'm making it probably more complicated than it needs to be, but you just have to understand and appreciate that, because when you're reading about terminology and where structures are, it'll really mess you up if you don't understand the basic kind of concept of this. Then ventral in the cerebrum is synonymous with inferior, okay? And rostral is synonymous with anterior. So just think about that, and when you have a free moment, relive it one more time. You can listen to the lecture again and kind of just draw it out and go, okay, I got it. It's no big deal. So when I'm reading about neuroanatomy, I got to know where I'm at, and what those terms mean based on where I'm at. We also have anatomical planes. Just like you have anatomical planes through the body, we have them through the nervous system. Okay, the coronal plane or the frontal plane, a sagittal plane down the middle, or a horizontal plane or transverse plane. Okay, so I'll show you some examples of that. So here's our different planes. So here is our, I thought I turned that feature off. This is the sagittal plane I'm kind of drawing there, is sagittal plane, and I'm kind of putting my scribbles over. Horizontal here is parallel with the floor, right here. Okay, and then the coronal here is this one here, kind of like that coronal or frontal view. Okay, sliced through that way. So here's your typical sections through a cerebrum. Okay, you'll see the first one here is the horizontal section. That's this one, like this, going this way, and I'm looking right down on the top there. I just cut the whole top of my cerebrum off. I'm looking down on what's left of it. Okay, that's a horizontal section. Coronal section, I thought I fixed that, but it's not seeming to work there. Coronal section, that's that frontal section. So it's like this, and I'm looking straight at the brain this way with the front of my brain cut off, is what you're seeing right there. Okay. Then this next one is a mid-sagittal section. That's as if I made a cut right down the center of my cerebral hemispheres, and I'm looking at the medial surface of that cerebral hemisphere. That's a mid-sagittal section. You could do parasagittal sections if you want off of that, but this is just a traditional mid-sagittal section. That's what you guys will have, as hemispheres that have been mid-sagittally cut through, right through the middle of the cerebral hemispheres, down this cerebral longitudinal fissure, that big, groove in the middle of your brain there. And then down below, that's a brainstem section, and that is a horizontal section through the brainstem. Okay. Most commonly, once you get to the brainstem and spinal cord, you only see horizontal sections. You just rarely see any other kind of section through that. They're mostly just going to see horizontal sections. Okay. More terminology. So let's talk about terms that are referring to a pathway or group of nerve fibers. So these are typically referring to axons that are traveling together, more than one axon traveling together. Okay, so these might make up, even a nerve. So you see one of those as a nerve. That's a group of bunch of axons that are together. Okay, but if you hear like a term like a capsule, okay, there's a big bundle of white matter in your cerebrum called the internal capsule. And you say, oh, that's just a bunch of axons. That's called the internal capsule. Okay, if you hear that term capsule, they're referring to a bundle of white matter. If you hear a radiation, that's also a bundle of axons. Okay, like we have the optic radiations. Okay, those are just axons that are taking information from your lateral geniculate nucleus back to your occipital lobe. Lemniscus. If you hear the term a lemniscus, it's like a lemniscus is ribbon-like. It's a flattened kind of bundle of axons. And within your brainstem, there's a pathway called the medial lemniscus, which is for proprioception from your body. As it goes up through the brainstem, it kind of flattens out like a ribbon, and it twists as it goes up. That's why it's called a lemniscus. Okay. If you hear a term called a tract, there's lots of tracts within our, the spinal thalamic tract that are running through our brainstem and our spinal cord. So if you hear the term tract, that's just, again, axons, a bunch of axons. If you hear the term fasciculus or funiculus, those are all axons. Okay, fasciculus means a little bundle or packet. A funiculus is a little larger than a fasciculus. So it's a bigger bundle or packet. Okay, and the nerve, or a commissural fiber, or commissure, a place where two things join. Okay. So we have what's called the anterior white commissure. We have the posterior commissure. These are structures that join different parts of the nervous system, like the two temporal lobes together, called the commissural fiber. But if you hear any of those terms, all we're talking about is axons or white matter. That's the main thing. If you see those attached to anything, that just means it's white matter axons. Okay? There's actually an entire textbook, it's like 600 pages long, that is on neuroanatomical terms and their alternatives. So for every term in the nervous system, there's like 14 other terms that can be used depending on if you're from the UK or from America, or there's just a variety of terminology that can be used. So nobody's kind of quite hashed it out to exactly what we want to use. The next kind of thing you want to know is what are terms that are used to describe groups of cell bodies. So the last terms were the axons. There's also some terms that are commonly used to describe a group of cell bodies that are going to give rise to these axons. So the names for these would be a ganglia, especially if it's in the peripheral nervous system. Typically, you refer to a ganglia if it's a group of cell bodies in the peripheral nervous system. Something like the dorsal root ganglia is in the peripheral nervous system. So a cervical ganglion is in the peripheral nervous system. There is one exception to that when we talk about the basal ganglia, which is a group of cell bodies deep in the cerebrum. That's in the central nervous system. And there's controversy. They think we should get rid of that term. We shouldn't call them basal ganglia anymore. We should call them basal nuclei. There are a lot of people that refer to them as basal nuclei because they refer to a nucleus as a group of cell bodies typically in the central nervous system. Okay, so if you hear that, if you hear a lamina is a group of cell bodies. We use that to refer to within the spinal cord. The gray matter is divided into these little layers called lamina. Those are different layers of different types of cell bodies. Okay, so if you hear ganglia, lamina, body, nucleus, just know you're talking about groups of cell. If that's attached to any other term, that's just a group of cell bodies. Any guys doing all right? Okay, I'm going kind of fast, but again, we're recording, so you can always go back. More terms. Furrows or depressions. So things that are like depressions or furrows within the central nervous system, or I shouldn't say so, the nervous system. We're usually talking about now our surface features. These are features that are on the surfaces of your nervous system, okay? A sulci, that's a rut or furrow, okay? So a sulci, and we look at the surface of the brain, it has lots of sulci, all those depressions. A fissure is larger than a sulci. It's a deeper kind of division, okay? And you'll see we have a lateral sylvian fissure, this big fissure right down the middle of our cerebrum, the longitudinal fissure, that's a deeper one than the sulci, okay? A fossa, if you see a fossa or depression, just like a fossa in a bone, we got fossas within our nervous system. Bumps and ridges, so there's also bumps and protrusions on the outside of our nervous system. So if you hear the term like a peduncle, that's a stem or stalk-like component. We have the cerebral peduncles that join our brainstem to our cerebrum. A gyrus, all the little bumps on the actual surface of the brain are called gyri or gyruses. And the depressions are called the sulci. These are the gyruses, the actual rounded bumps. A brachium, we have a brachium of the inferior colliculus and blah, blah, blah. But that's another thing. It's just a bump on the outside of the nervous system. A tubercle, just like a tubercle on a bone. We have tubercles on our nervous system. There's little small nodules or small eminences. And then we have layers within the nervous system. So if you hear the term a lamina, cortex, tectum, or tegmentum, those are describing different layers within the nervous system and different regions of the nervous system. So if you get to that, you see that come up in a textbook or something, you're reading, you go, oh, that's just describing a layer. We have the cortex on the surface of our brain. We have the tectum, which is the roof of the midbrain. Well, I'll show you that when we get to the brainstem. Okay. All right. Now we got some terminology out of the way. Now we're moving into the actual anatomy and getting into that. Okay, and we'll try to keep it fairly, concise and functional. And we're going to start with the big guy, the cerebrum. Cerebral hemispheres. What does it do? Before you know anything else, just know basic functions of these things. Just really understand the basic processes. Cerebral hemispheres do a lot. They do sensory. They process sensory information. They help create motor signals. You use memory, reasoning, language, intelligence, personality, emotion. This is where it all happens, as far as the cerebral hemispheres for making you, okay? It has four primary lobes, okay? A frontal, parietal, temporal, occipital lobe. There's also a fifth lobe, okay, that most people don't talk about, although you may see it pop up, called the limbic or insular lobes, and I'll show you where that is in a second on a picture. There's a longitudinal fissure, this big, deep fissure that divides it into two hemispheres, okay? There's a corpus callosum. There's this this connecting commissural fiber that connects one half of your brain to the other, called the corpus callosum. And if you take and you like pull open that longitudinal fissure and look right down, you'll see this nice white shiny thing. That's the corpus callosum. It's a bunch of axons connecting one half of your brain with the other, so you can get communication between the two hemispheres. The surface is composed of gray matter, just a few millimeters thick. That's where the cell bodies are, mostly in the cerebrum. All the cell bodies are in that little few millimeter thick layer of cortex. You do have a few deep nuclei, the basal ganglia or basal nuclei that are down in there, that are stuck in there. And that's made up of what's called the caudate, globus pallidus, and putamen. We'll point those out in a few minutes. And then the diencephalon, which is also cell bodies. It's a group of nuclei, the diencephalon, which is our thalamus and hypothalamus together, essentially. So that's really the cerebrum overview, is what's in there, what it does, and then some of its major parts and components. Let's look at pictures. Pictures are better. Oh, in a second, we'll get to pictures. The basal ganglia I've mentioned. Those are sitting down in the cerebral hemispheres. They're a group of nuclei associated with many functions. They help to process movement, vision, cognition. We talk about them in PT mainly for their role they play like in things like Parkinson's disease. The basal ganglia circuitry is really important for helping you move normally and express movement normally. Because when you send a signal down to go tell your muscles what to do voluntarily, it doesn't go straight to the muscle. It actually goes through the basal ganglia first. There's a little loop through the basal ganglia before it goes back up and releases the signal. If it gets hung up in that loop, it slows down, then you get Parkinson's disease. Okay, if they can't get through that little basal ganglia. But you have the same thing for voluntary vision when you like try to look to different places. Even memory and intellectual kind of things and processing, some of that loops through the basal ganglia. So basal ganglia problems, like people with Parkinson's, can also have cognitive problems because it's a relay center that helps with that. The diencephalon, it's a major relay center for sensory, motor, homeostasis, reproduction, eating, and growth. It also provides the control for the central nervous system, autonomic nervous system, the master regulator via the hypothalamus, okay? So it does a lot. The thalamus itself, the hypothalamus and the thalamus are the two main things that make up the diencephalon. Some books will also include the epithalamus and the subthalamus as part of the diencephalon. Some don't. Anything with an alamus on the end, together could be considered part of the diencephalon, but di is two, so really they're talking mainly the thalamus and hypothalamus. They make the diencephalon proper, we call it. But the thalamus is the egg-shaped structure in the middle. We'll show you that. It's a relay structure. The thalamus is like the secretary of the brain. If you want to go talk to the cerebral cortex, you got to go through the thalamus first. So any sensory information coming in, especially, has to go through the thalamus. Or if you're going to go into the basal ganglia and you want to go back up to the cortex to tell it some information, you got to go through the thalamus. So the thalamus is a relay structure. It's kind of like this, kind of sifting, sorting system for the brain. You got to go through the thalamus first before you go talk to the cortex. The hypothalamus, that's that autonomic nervous system regulator, okay? Eating, reproduction, homeostasis, all driven by the hypothalamus. Epithalamus, that's also referred to as the pineal gland. It helps to regulate the pituitary and the adrenals, okay? So it plays a role in some homeostasis functions. The subthalamus is actually part of the basal ganglia circuitry. So it's a little bit of a kind of a, that does its own thing, but it's part of the basal ganglia. It influences the basal ganglia circuitry a little bit. Don't worry about those too much, okay? All I want you to appreciate from this picture is that you've got these paired structures on both sides. Or deep within the cerebral hemispheres, you have, I'm going to outline this structure right here. This is primarily our basal ganglia structures in purple. Just medial to that is these two egg-shaped structures. These are our thalamus. And then below that is the hypothalamus, kind of sticking out below that is the hypothalamus, okay? Amygdala is part of the limbic system. It's actually not part of the basal ganglia. It just happens to be at the end of little tip of the tail of the caudate there. But so just appreciation. These are paired structures on both sides of the brain. You got basal ganglia on both sides, a thalamus on both sides, and they're paired up. Okay, and those are all cell bodies in there, nuclei. Okay. All I want to show here is this is that diencephalon here, but this part right here is the thalamus right here. So this is the thalamus, and then this is more the hypothalamus just down below it. And a little anterior and inferior is the hypothalamus part of that. Okay. diencephalon. Still on cerebral hemispheres, or in cerebrum here, we have the different lobes. We have the frontal lobe right here in blue. What's it do? Again, think basic. Motor and personality. That's motor and personality is the frontal lobe. Temporal lobe right here, light orange. What happens there? Memory. Your hippocampus is deep inside of there, and that's where you get initial auditory processing. It makes sense that the temporal lobe kind of sits where your ear is, so it makes sense that auditory information would go there first, right? Then the parietal lobe, back here, it is processing a lot of sensory information, but it's also a huge integrative associative area. If you'll notice here, and there's your occipital lobe sitting back here, which is primarily vision, but you'll notice the parietal lobe, it has borders with the frontal, temporal, and occipital lobes. So the parietal lobe is very important for integrating sensory, visual, auditory, and even motor actions all are integrated and stored in there and made sense of. That's the parietal lobe makes sense of all the information coming in and helps you make sense of the world. Okay, so. And then the last lobe, that fifth lobe, is sometimes referred to as the insular lobe. Okay, if you take and there's this lateral fissure, also known as the Sylvian fissure. If you pull it apart with your fingers or from some forceps, if you want to try it on one of these brains, just be a little careful. If you tear it up a little bit, that's okay. Pull that apart, you'll see some more gyri and sulci a little deep in there, and that's the insular lobe. Okay, sometimes called the limbic lobe or the insular lobe, and so it has some more limbic system function, which is associated more with emotions and those kind of things, and a little more autonomic regulation associated with emotions. Okay, so that's the insular lobe. I'm not going to go through this whole slide. I just think it's a good representation to say, this is a left hemisphere, okay? So it's a left hemisphere. It's important to know this is a left hemisphere because left hemisphere is where you have more language function, okay? At least like verbal language and written language. The right hemisphere is for nonverbal communication, okay? Interpreting what other people are, their nonverbal communication and also your nonverbal communication. So, which actually about 80% of our communication is nonverbal, 80-90%. So the right side is also important for communication. When you think about speech and reading and writing and speech, that's the left side. So when you look at some of these areas that they outline, some of those are more related to the to actual spoken and written type languages on this left side. This area right here that's outlined in dashes, on the left side, that's where you interpret spoken language. So you make sense. You notice it kind of comes up into that parietal lobe region. That's where you make sense of somebody talking to you and say, oh, they're communicating. They're saying words, and the words mean this. That's where that happens in that Wernicke's area. When you are trying to talk to somebody and express your own words, or even in writing as well, you use this area right here. This area is Broca's area. Okay, Broca's area is used for expressing language. Wernicke's area is reception of language. Okay, and so you may in the clinic or something, if somebody's had a stroke and the stroke affects Wernicke's area, it's called receptive aphasia or Wernicke's aphasia. They can't understand what you're saying to them, but they may be able to still speak. However, it'll still sound not quite right because they can't check themselves. They can't understand what you're saying, and they can't really understand what they're saying, even though the words come out. It's sometimes called fluent aphasia too. Is they'll just start rambling, beautifully sounding words, and you think they're making sense, and all of a sudden you realize this is not making sense because they can't understand and check that they're making sense. If you have a damage to that Broca's area farther forward, they can't talk. They can't find the words. They can't express themselves, but they can understand. And they get really frustrated because they know that they can't get the words out right because Wernicke's area is okay, and so they're going, crap. And then the more frustrated they get, the harder time they sometimes have getting it out. And they're tough patients to work with because you got to be patient and give them some time to try to get it out too. But even though we got exercise to do, we got stuff to do in the clinic here. But that's Broca's area up front. So just to get an appreciation for some of the more specific functions at some point. This is that coronal section. You know, this is a coronal section through the cerebral hemispheres. And the main things I just want you to see here, a couple of major things. This is basal ganglia structure. This is a basal ganglia structure. This is also part of the basal ganglia here. I don't know why they have that colored in green. It should match these guys. This is called the caudate. Caudate. This right here, it's actually multiple pieces together. This part out here is called the putamen. There's another little segment here called the globus pallidus internus, and there's another segment here called this is the globus pallidus externus, and the most medial segment there is the globus pallidus internus. Okay, this down here is the tail of the caudate. This is the body of the caudate up here. The one down here is the tail of the caudate. Because that caudate, remember that it's like C-shaped, it wraps all the way around and down. So then this right here, this green structure here, and again, everything's paired, that's your thalamus. Okay, that's your thalamus right there. That's all I want you to see from that. It's just we're kind of how those basal ganglia structures are sitting in there with the body of the caudate here. This is the putamen. This is globus pallidus. I can't write on this thing. I wish the screen. This is globus pallidus externus, and this is globus pallidus internus. Okay. And then tail of the caudate sitting down below, coronal section. You can kind of see this picture shows you where on top there shows you where that cut is taking place. Okay, and you're looking straight in. I don't know why that thing did not take that. Change that, man. There we go. Hang with me. We're doing good here. We're moving down to the brainstem. Okay, so that was cerebrum. Any questions on the cerebrum? Okay. And we'll get to look at cerebrums again here, the real stuff, and go through some of those same things. So, brainstem, three main parts: midbrain, pons, medulla. We're looking at the front of the brainstem here. There's the midbrain. This section here is the pons, and then below that is the medulla. Okay? We're going to point out just some major structures, external features, okay, on the front part of the ventral surface of the brainstem. So this would be the anterior or ventral surface of the brainstem. Okay, they are not on here, but you would see if they hadn't been all torn off. This is a nice clean one. They've taken all the cranial nerves off, just kind of shaved them all off of there, pulled them all off so they don't distract you. But your cranial nerves 3 through 12, you could appreciate on the front of the brainstem. Cranial nerves 1 and 2 are coming up from your cerebrum, your optic nerve and your olfactory nerve. Those are coming out of your cerebrum. That's why you don't see that. You start cranial nerve number 3 through 12 down here in the brainstem. Okay. From the front, this right here, this is called the cerebral peduncles. These guys right here. Remember, peduncles it's a connecting part, and there's just a bunch of axons in there. Peduncle is a bunch of white matter axons, and you can actually see kind of some of the striations in that from the front. If I erase that, you can see some of the striations there. That's just showing these are axonal fibers running up here, and this is actually where quite a bit of your motor fibers are coming down through here, down the front of your brainstem, coming from your cerebrum. There's a bunch of motor fibers that run in that front part of those cerebral peduncles. This cave right here, this nice dark cave here, that's the interpeduncular fossa. A fossa is a depression. It's a depression in there, just a space, okay, between the two peduncles. These are not that important, but people always wonder what these are. These are called the mammillary bodies, and you can probably appreciate the mammillary nature, the term used to describe those, by the way they look, but they are more autonomic in nature. They don't have they're not even sure exactly what the mammillary bodies do per se, but they think it has some influence over some autonomic function. The basilar pons is just this whole front part of the pons. This bulbous front part is called the basilar pons, that rounded front part of the pons. And then there's a groove in here. It's kind of subtle, but there's a basilar groove. They call it the basilar groove because your basilar artery lies within the basilar groove, okay? And it's part of your basilar pons. So there's a little groove there that basilar artery runs in when we get to some blood supply. Now down on the front of the medulla, we have what's called the pyramids, which is this area right here. They're kind of pyramidal in shape, triangular in shape. But then I also forgot, I should have put on here, oh, I forgot, I skipped the olives. The olives are right here. Those little bumps right out there, okay? So just don't get those confused. The pyramids are a little more medial, and then the olives are a little bigger bumps more lateral there, okay? All of the olives, what's deep in the olives, if you go inside of there, is called the olivary nuclei. And those have connections with their cerebellum. That's all you got to know right now. They help with coordination, okay? These pyramids right here, you actually have your motor fibers run down through the front part of the brainstem here, and they run down through the pyramids there, okay, down the front part of the brainstem, the motor fibers, okay. Before, right down here, they're called the pyramidal decussation. They start crossing over to go to the other side, because remember, your left side of your brain controls the right side of your body. From a motor standpoint, it feels, the sensory is also crossed over. And so there's an area right down here where these fibers start to cross over, and that's called the pyramidal decussation, okay. So the pyramids cross or decussate there. I'm going to go back out here and see if I can somehow get that transition. Maybe I forgot to. Okay, I clicked that off. Should be okay now. So pyramidal decussation right there. If you had a little probe and you start, like if you have a brainstem and you take a little probe or some little pick thing or something, you start sliding it down the front in that, there's a little groove there in the middle there, that ventromedian groove there. You start dragging it down, all of a sudden it'll stop. And that's where the decussation starts. That's when those fibers start crossing, those motor fibers start crossing, and you'll kind of feel that stopping point there. Back part of the brainstem. A little more to look at here. We have 4 bumps right here. There's a bump, there's a bump, there's a bump, and there's a bump. The 2 bumps on top are called the superior colliculi. The bumps on the bottom are called the inferior colliculi. Okay, so superior and inferior colliculus. The 4 bumps together are called the corpora quadrigemina, which is 4 bodies. So corpora is body, quadrigemina, 4, corpora quadrigemina. So, but all you have to know about those is those are 4 bumps on the back of the midbrain, okay? And they're relay centers for reflex movements associated with hearing and vision. So when you, if the superior colliculi, your eyes, maybe you think of, I think of my eyes a little higher than my ears. Maybe they're not, but a little bit higher. So the superior colliculi are for vision. So when you see something moving in this, that comes into view, or somebody walks into a room, and you know how it's just so hard not to look. You got to kind of look. What happens is once that visual image hits your eye, before it even gets to your occipital lobe, it goes to that superior colliculus, and the superior colliculus sends signals to your neck muscles to tell you to look to that, to see what it is, make sure it's not a threat, something like that. It's a built-in, hardwired kind of reflex that you kind of have to. I mean, you can not do it, but you have to consciously overcome it, to do that. You have the same thing for hearing. If you hear a sound, before it even gets to your temporal lobe and you register it as a sound, it goes to that inferior colliculus, the lower one, that also connects to your neck muscles and tells you to look towards that sound, okay, and turn your head towards that sound. So that's what those are for. This is interesting. There's only one cranial nerve that comes off the back of the brainstem. All the other cranial nerves come off the front or the side of the brainstem. There's just one that comes off the back of the brainstem. And that's the trochlear nerve, okay, that's this guy right here. And it goes to one little muscle in your eyeball called the superior oblique. Don't need to write that down, but just it goes to, it's cranial nerve number 4, okay, so that's and it's coming off this back part of the brainstem right here, right below that little inferior colliculus on each side. There it is, coming off there. What else do we got back here? We've got a thing called a rhomboid fossa. Remember, fossa is just a depression. This is kind of rhomboid in shape. basically what we've done, the cerebellum sits back off the back here of this area. And we've cut the cerebellum off, so you can see the back of the brainstem there, okay? And then when you take that cerebellum off, you see that rhomboid fossa sitting there. And that rhomboid fossa, it forms basically, we call it the floor of the fourth ventricle. There's a space in there, and there's cerebrospinal fluid in there, and your cerebellum, when it's attached, it forms the roof. And so when you have the cerebellum and it's stuck on the back, there's a little space in there between the cerebellum and that rhomboid fossa, and that's where cerebrospinal fluid's inside of there. That makes up the fourth ventricle. Okay, so the rhomboid fossa is kind of like the floor, or if you're standing up like this, it's really kind of like the front of the fourth ventricle. The cerebellum is the back of the fourth ventricle, but they refer to it as the floor of the fourth ventricle. But really, if you're standing upright, it's the front or the anterior surface of the fourth ventricle. Okay. Here's where the cerebellum's been cut off. I'll show you the cut points. This whole region right here, that's where the cerebellum, you can see where it's been cut off the back of that, okay? And we've cut it in a way that you can appreciate some different parts of that cut, okay? There's actually some connecting parts to the cerebellum. One is called the middle cerebellar peduncle. Remember, a peduncle is a connecting stalk. So the middle one here connects the cerebellum to the brainstem. There's one here. This one here is the superior cerebellar peduncle. And then there's one down here called the inferior cerebellar peduncle. Okay? On this side, they've cut it a little differently. Here's your superior cerebellar peduncle. This region right here is your inferior cerebellar peduncle. This is your middle cerebellar peduncle out here. And those are just ways that axons go in and out of the cerebellum. So when you have like little muscle spindles and receptors in your joints and so forth, they get into the cerebellum by coming up from down low through here. They come into the inferior cerebellar peduncle to get into the cerebellum. Okay? When you want to go up and tell the brain to make an adjustment based on some of that sensory input, you actually send a signal up through the superior cerebellar peduncle to go up. Okay. You also send copies of your motor messages from when you actually go to do an activity. You send a copy to your cerebellum so it can compare what actually happened. Those come down from the one side of your cerebrum. They come into the middle cerebellar peduncle to go send a copy to the cerebellum. So then you can make a comparison between what's coming in to your cerebellum to what you intended to do. But again, don't worry about those details. Just giving you some real basic clinical reasons why you want to know this stuff. Okay. Back here on the, now we're down. So that was the back. So this is kind of the midbrain at the top. This is, I don't know why that change is not, maybe I need to resave it with that change. This is the pons right there, and now everything below that line is the medulla. Okay. And only a couple things we'll point out in medulla. We have these little tubercles or little bumps, and you almost have to kind of really imagine them, but there's a little bump right there, there's a little bump right there, a little bump right there, a little bump right there. The ones that are more medial are called the gracile tubercles. So these little guys, the most medial ones right here, gracile tubercles, and the more lateral ones are the cuneate tubercles. Okay. These are associated with is when you, for sensor information, proprioceptive information, information telling you about your body's position in space that you consciously are aware of, it's coming up the back part of your brainstem. The ones that come up to this little area are coming up what's called these are called the dorsal columns as a group, but this is the fasciculus, this little pathway here is fasciculus gracilis, and it's taking axons up to this little tubercle there, which just below that is the nucleus gracilis. Once it makes a connection there, it crosses over the other side and goes up in your brainstem to send the signal to the opposite side of the brain. Okay? And then that's for the lower body. The upper body comes up what's called the fasciculus cuneatus, and then makes a connection to another cell body within the nucleus cuneatus, which is just below that tubercle, just underneath that tubercle, and it does the same thing. It crosses over and then goes up to the opposite side once it's made that connection. Okay, don't get overwhelmed by the detail here, just appreciating why these structures are even relevant or why I'm pointing them out. Let's get to the cerebellum, then we'll take a short break. So sitting off the back of the brain, what we just talked about, showed you how it was cut off the back of the brain stem, is this thing called the cerebellum. Just appreciate looking at that, all the little folds and sulci and invaginations that are in that, how wrinkled that thing is. The more wrinkles something has, the more it's trying to take advantage of surface area. You know, if you keep folding in on yourself, you've got more surface area. And just like your cerebrum, all the cell bodies for the cerebellum are on the outside, on that surface area. And so the cerebellum is incredibly important for processing all the proprioceptive information coming in from your body. It's unconscious. This is, you're not consciously aware of any of the information going to your cerebellum. It's all happening without your conscious awareness. And it's making corrections on the fly without you thinking about it. Okay, it's very important for motor learning. Okay, as you start to learn a new task, and your cerebellum is trying to like figure out, and you're making errors, it happens. It's sensing those errors and making small corrections with each successive trial, and then hardwiring that so that you can do it without thinking about it. You know, so that's when you kind of get to that point where you're totally independent and not thinking about any of the movement you're making. Automatic type movement is what the cerebellum is really important for. So. Yeah, it's insane how much processing that takes to do that, for that to occur. We have an anterior lobe and a posterior lobe. Let me maybe go with a little lighter color here. This right here, that's your anterior lobe. Then all the rest of this back here is your posterior lobe. We mainly identify the anterior and posterior lobe looking at it from this this is a sagittal cut through that, okay? We can, this picture on the lower right is a superior view looking at the top of the cerebellum. So the cerebrum's gone, so you're just looking at the top of the cerebellum. There's a line right there. It's kind of hard to appreciate, but that's your anterior lobe, then all the rest of this is your posterior lobe. So it's a little harder to define exactly where those anterior and posterior lobes are looking at it from, you know, from the top here. Okay, so if we were looking at it down like this from the top here, a little harder to see where that pickup, where that space is there. The cerebellum can also be divided into these longitudinal divisions. Okay, and it's probably best appreciated, those longitudinal divisions. This is looking at the front. The one on your lower left is looking at the front of the cerebellum. So looking at it this way as it's been cut off. Okay. The very middle section right here, if you're reading something about cerebellum, they're talking about the vermis. They're talking about that very middle section in a longitudinal section is the vermis. Okay, vermis means kind of worm-like, and it kind of looks like, if you get rid of something, it looks kind of like a little bit of a worm. I don't know. Then right out here you have what's called the paravermal region. So the paravermal regions is just a little bit more to the outside, and then these are their lateral hemispheres out here. And believe it or not, your body is kind of somatotopically arranged in this longitudinal feature. So the vermal region, it helps coordinate the muscles for like your mouth and your eyes and the things that are more midline. As you get to the paravermal region, that's more like your girdle muscles and some of your trunk muscles for coordination of those. And you get to the lateral hemispheres, that's the coordination for your fingers, your toes, your hands, more lateral. So it makes sense that from medial to lateral, it's kind of like your body. It's more trunk in the middle and face and eyes and nose. And then as you move more laterally, it gets more out to the outside of your body as far as what those regions are talking to and helping to coordinate. So from a functional standpoint, it's more important to know. So I have a patient who has a stroke in the lateral hemisphere. I'm going to expect a lot of incoordination with their distal muscles. But if they have a stroke in the middle of the cerebellum, they might have a lot of truncal ataxia, or they have ataxia of their speech, where they can really affect, or their eyes are jumping around because it's got that middle vermal region that causes nystagmus, we call that. And then lastly, just the cerebellar peduncles. Remember we showed you where those peduncles were on the back of the brainstem, where those cuts were? We have the same thing here. This is the middle cerebellar peduncle. This would be the inferior. This would be the superior. Okay, middle, inferior, and superior cerebellar peduncles. Okay. Let's pause there for a moment. Any questions? Whew, this is not the right time to be doing this. There is never a good time to do neuroanatomy. At least you don't have distractions like I did. When I went to physical therapy school, I sat in a lab. I was at Loma Linda University in Southern California, and my lab, you'd open up the door, and we'd a lot of times leave it open because it's always beautiful outside, just let the air in. I'd look over my shoulder, and I could see the San Bernardino Mountains, and they'd be like covered in snow, and I'm like, I wonder what the skiing conditions are like. And then I'd feel the wind. If the wind was blowing a certain way, it would blow into the lab, and that means it's offshore conditions. I'm like, oh, I wonder what the surf is like. So it's like, I need to get out here. I got to either surfing or skiing. I don't know which I'm going to do today, but that was the choices. That was the hard decisions I had to make each day after class. You guys don't have to worry about that stuff here in Ohio. Just all you got to do is like watch Game of Thrones or something like that. Yeah, right. So now we're moving to the spinal cord. What is it? The spinal cord lies within the vertebral canal of the vertebral column, nice and protected in there for the most part. Extends to the level of the L1, L2 intervertebral disc. It doesn't go all the way down. It stops actually pretty high up, you know, and so just remember the spinal cord doesn't go all the way down. That's important to know because if it stops up high there, then you can do things like put needles down in below that level so you can get cerebral spinal fluid or give somebody an epidural injection and those kind of things without risk of hitting the spinal cord. It has a thing called the cauda equina, which is at the end of the spinal cord where, since the spinal cord ends at L1, L2, but you still have nerve roots that have to go all the way down to the sacral nerve roots. They kind of extend down and hang down off the end of it. They look like a tail, so it's the cauda equina, horse's tail. That's just the nerve roots coming off below that area of L1, L2 and just kind of hanging down off the tip. There's a thing called a filum terminale, which is a little extension of a covering of your nervous system called pia. And pia extends off the tip of the spinal cord and goes all the way down and anchors it to the sacrum. Okay, it's this little tiny, you know, on the surface of your spinal cord, this pia is microscopic. You can't even see it. But when it comes and extends off the tip, there's enough of them there, the cells, they actually form a little strand that anchors your sacrum to your spinal cord. You have these things called dorsal roots and ventral roots in the spinal cord. So when the spinal nerves come off, you have a dorsal and ventral root that go to form that spinal nerve, and I'll show you this. The one in the back, the dorsal, is afferent, brings information in. Sensor information comes in the back part of the spinal cord to the dorsal roots. Afferent information, motor, autonomic, going out the nerve is going to go out the front part and go in through the ventral roots. Okay. You have 31 pairs of spinal nerves, okay, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. Okay. So here, just show you a few things with that. Here's a sagittal section through the spinal cord, that one on the left, and here's the tip right down here, right between that L1, L2. Region here is where it's ending here, the disc space between L1 and L2. All the rest of the stuff coming off, these little nerve roots coming off, those are the that's the cauda equina. Okay. Darn it. So this gives you can see also. You notice how there's quite a bit of space down in here. I'm going to go back to a little darker color. Yeah, I don't want that. There's quite a bit of space back here in the lumbar spine too. So there's room, like if you stick a needle in there, hopefully the nerve roots move out of the way, you know, and can float out of the way. But occasionally you nick one, you know, like because they're doing, a lumbar puncture or doing an epidural or something like that, and it'll irritate it or nick it, and it might hurt for months. You know, if you really, you know, tweak that nerve root and damage the myelin that surrounds it, it can take a while to remyelinate and not have pain there and so forth. So there are some risks, but most of the time it moves out of the way when you do that. Okay. Much tighter up in the cervical spine. You can see there's not much space up in the cervical spine. So here is the tip down here too. That's the tip of the spinal cord right there. And this is all that cauda equina. And then there's a little tiny thing here. I'm afraid if I go back out and try to resave this thing, it'll screw up my recording. So that's why I'm not going back and trying to. There's a thing that comes off the tip here. There's a dark purple thing that's coming down. That's the phylum terminale, and it attaches to the sacral bone. You really can't see it in there, but there is actually a little tiny purple one in there. You can see in the middle there, a little tiny purple phylum terminale. One other thing to appreciate, too, is that the nerve roots in the cervical spine, they come out above the vertebrae of their vertebral level. So C1 is coming out above the C1 vertebrae, okay? When you get down to C7, then you have a C8, but there is no C8 vertebrae. And so at that point, C8 comes out below the C7, and then from then on, since there is no C8, the next one is T1. So in basically the thoracic spine and lumbar spine, the nerve roots come out below the vertebral level. The nerve roots are exiting below the vertebral level, where in the cervical spine, until you get to C8, they're exiting above the vertebral level. So that just means if you have a disc problem, you know, in the cervical spine versus the thoracic or lumbar spine, it's important for knowing what nerve root it might push on, you know, and cause compression of. Okay, so if you're, this is going to really bother me. If you have a bulging disc between C1 and C2, it's going to push on the C2 nerve root that's exiting above that C2. Okay, if this is a bulging disc between C1 and C2, we're down the thoracic spine. If you're between if you're between T1 and T2, here's T1 and T2, it's going to press on T1, not T2, okay, just because of the way it exits. So just that's all you need to appreciate, that is that when you're looking at an MRI or you get a report or something like that, if they say the person has a bulging disc at this particular level, if it's down in the lumbar spine, you know, it's going to be the one. So here, if it's between T1 and T2, it's going to be associated with the higher level. It's always they're always going to give you between T1 and T2. It's going to cause a problem with the T1 nerve root, versus with the cervical spine, if it's between like C1 and C2, it's going to cause a problem with the C2 nerve root. Okay, so it's the one that's the lower vertebrae in the cervical spine, but in the thoracic and lumbar spine, it's associated with the nerve root of the higher vertebrae, the most, you know, proximal or the most caudal vertebrae. So anyway, I'm sure might see some sensory or motor impairment if it's compressing that nerve. Now, this is looking at a horizontal section through the spinal cord and the internal features. Different from the cerebrum, where most of the gray matter is on the surface of your cerebrum and cerebellum, the cell bodies, spinal cord, the cell bodies are all in the middle. Okay, so the gray matter is in the middle. All the white matter that is axons are on the outside. Okay, and so you have this H-looking structure, which contains a dorsal horn in the back. You see a dorsal horn on each side, these dorsal horns. Okay, you have ventral horns. You also, in at least the thoracolumbar region where the autonomic sympathetic outflow is, you also have these little lateral horns, because that's where the cell bodies lie for the autonomic nerves, okay, that go out. So they're harder to see, but just so if you see that, that's a little lateral horn. But you don't have that in the cervical or lumbar spine. You only see lateral horns in the thoracolumbar region where those sympathetic nerves are coming out. You do see a few lateral horns down in those few sacral segments where those parasympathetic neurons are as well for the parasympathetic nervous system. But for the most part, you see just the dorsal and ventral horns. Okay. There is a central canal. There's actually a little canal there that's in the middle there. That's called the central canal. The white matter is mainly axons, and you have both ascending and descending tracts all in these different regions. This whole area back here, that's a big area of axons called the posterior funiculus. This area here is called the anterior funiculus. These big areas here, those are the lateral funiculus, okay, on each side. And within those funiculi, there's lots of different tracts and subparts and smaller components, which you'll see in a few minutes. Here's just an appreciation of how the spinal cord lies within your vertebral canal. okay? And so there it is, right there, lying in the center. Here is the dorsal root coming off the back and the ventral root coming off the front, okay? And so dorsal root, this is where information goes into the spinal cord on the back, goes into through the back door, okay, as far as the sensory information. Pain, temperature, proprioception, all those kind of things come in through the back part of the spinal cord before they go up, okay, before they go up the spinal cord, okay? The anterior root, this is where your motor neurons send signals, and if there's an autonomic, if this is like in the thoracolumbar spine, autonomic neurons also go out the front part of the spinal cord to go to innervate muscles, smooth muscle, glands, sweat glands, that kind of stuff. That's going out the front, and so that's efferent. That's an efferent kind of exiting information that goes out. The efferent axons go out the front. The afferent axons come in the back. Okay. This little kind of larger enlarged area, actually what happens when most of your information comes in for your sensory information, it makes a synapse right here in this thing called the dorsal root ganglion before it goes into the spinal cord, okay? So that's just the dorsal root ganglion. Remember we said a ganglion is a group of cell bodies out in the peripheral nervous system, so that's a dorsal root ganglion, okay? And then once they come together, this is your spinal nerve right here. Very quickly, that spinal nerve divides into a dorsal ramus. This is where all the information goes to the back side of your body, like along your paraspinals and the back part of your skin and everything goes out the dorsal ramus. And then the ventral ramus, this guy goes out, it swings around and goes to the front part of your body. So supplying all the front part of your body, muscles, skin, all that kind of stuff, sensory, motor, all goes out through that ventral ramus. Because everything, once you get to the spinal nerve, everything's together out here. You have motor, autonomic, and sensory axons all together. It's not here till they start to split apart, okay? Don't worry about the rami communicans. That's actually has to do with autonomic function. It's actually, when you send autonomic neurons out, the sympathetic neurons, they go through this little guy here. They go through a little loop first before they go out. There's a little ganglion that sits on the side of the body called the paravertebral ganglia. Again, it's where cell bodies for autonomic function, sympathetic nerves make their first synapse. Don't get too hung up on that, but that's just what that is. That was spinal cord. All right, so we've covered a lot of the main components. Now we're going to talk about some of the support systems for the nervous system. This includes the coverings of the nervous system and the blood supply. So we're going to talk about coverings of the nervous system, okay? Collectively, these coverings of our nervous system, and we're talking really the central nervous system here, okay? This coverings of the central nervous system, are called the meninges, okay? These are protective membranes of the brain, brainstem, and spinal cord, okay? And there's 3 of them. They're called the meninges, plural, because there's 3 of these meninges, these 3 layers, okay? And the outermost layer is called the dura mater. It forms some important structures that divide your brain and also divides between your brain and your cerebellum and so forth, and also forms some of the tubes that some of your venous blood gets out to drain from your brain, goes through these drainage ditches that are formed by dura, okay? So we have the falx cerebri. The falx cerebri, your covering of your brain, dips down in that fissure between your 2 cerebral hemispheres. It's called the falx cerebri. It's diving down between, it's called the falx cerebri. The tentorium cerebelli, it's like a tent, and it's like this dura mater goes over the top of your cerebellum, between the bottom of your cerebrum and the top of your cerebellum, and kind of forms a tent over the top of your cerebellum. So if you hear, like, if you're reading an MRI or you read a report and a person has a supratentorial lesion, that means they have like a bleed or a tumor that's above the tentorium cerebelli. Basically, it just means it's above the cerebellum. If they have an infratentorial lesion, that means it's in the cerebellum or the brainstem. It's below that tentorium cerebellum. So if you hear they have infratentorial bleeding or supratentorial, that's all that's referring to. And that's why some of these terms are just nice to know for your own, kind of understanding. The dural venous sinuses, these are these little drainage ditches that are formed by the dura that eventually your venous drainage from your cerebrum goes into those and drains out. And we'll show you that when we get to the blood supply. The arachnoid is the next deepest layer. Arachnoid, spider-like, and actually one of these brains, there's a lot of the arachnoid left on. Most of these we've taken the arachnoid off, but one of them there's quite a bit of arachnoid left. It looks very spiderweb looking, kind of look like, and right below that right below the arachnoid, that's what we call the subarachnoid space, and that's where your cerebrospinal fluid actually flows. It's between the arachnoid and the pia. The pia is the most intimate layer, and it's just microscopic, and it's right on top of the nervous system tissue. Then there's a little gap between the pia and that arachnoid, and you really need to have cerebrospinal fluid to have a gap there. It's a potential space because you need some cerebrospinal fluid to puff up the arachnoid, so there's some space there for the cerebrospinal fluid running through there, okay? And then your dura is the most outer layer. If you go from inside to out, you'll see that pia, the P in pia, the A in arachnoid, and the D say pad. It's the padding of the nervous system, but if you just memorize from inside to outside, it goes pia, arachnoid, dura, pad, forms a pad for your nervous system. Question?
Is there a difference between subarachnoid space and like subdural space? Are they the same thing? Or?
We're going to show, yeah, that's a great question. Actually, I'm going to show you about subarachnoid bleeding versus subdural bleeding and show you some pictures of that, so that'll make it clear. But yes, there is a difference. So here's like a little cross section through this is kind of like a little horizontal section through the top of the brain. You'll see where the skin and bone are there. And then they're really tight together, but that outer layer, that pink layer is the dura, really tough. It's leather-like. It's crazy how tough it is when you cut through it and feel it. It's kind of shiny. But then here's the arachnoid, and it's got these little kind of end feet that protrude down. But there's space in there. There's space for cerebrospinal fluid to flow between that and this pia, the real microscopic layer right on the surface of the cerebral cortex. Okay, and you'll notice that the most of your blood vessels that are on the outer surface of your cerebrum, they are in the subarachnoid space. Okay, so when somebody says somebody has a subarachnoid bleed, that means one of the blood vessels that is in that space is bleeding between the arachnoid and the pia. It's subarachnoid. It's just below the arachnoid. And that's very common to have a subarachnoid bleed because that's where a lot of your blood vessels on the surface of your brain are in that subarachnoid space. Okay. So this picture here, it's a little hard to appreciate, but normally what is happening here, let's see. Normally, this is all dura covering the whole surface of your brain. What we've done is cut the dura off and just left where the dura dips down in between. This is looking at the back of the brain, the back of the cerebellum. You can see the dura folds in, falx cerebri, it kind of folds in between the cerebral hemispheres, but then you can see it also dives in between the bottom of the cerebral hemispheres and the top of the cerebellum to form that tent. Okay, and it is kind of tent shaped because it kind of does one of these. It kind of goes up like this. So this is showing you that same thing in a sagittal section now, but this they're showing you the membrane here. This is the falx cerebri, the dura that's dropped down to dive in between the two cerebral hemispheres. And this is actually the tentorium. This is the tentorium cerebelli diving up in here between the bottom of the occipital lobe and the cerebellum sitting back here. Okay, so everything below, this is the tentorium coming up to here. This is all the infratentorial space. Is all this area right here is infratentorial. Everything above it is supratentorial. Okay. Blood supply. So we got to get some blood up to this brain to make it function. Two major supplies of blood that go to the cerebrum, to your brain and brainstem. One is via the internal carotid arteries. Okay, the internal carotids provide most of the blood to the cerebrum. Okay. The internal carotids, as they're coming up to supply the cerebrum, divide into two major vessels. They divide into the middle and anterior cerebral arteries. Okay. That's going to supply most of the outer surface of your cerebrum and also the inner surface of your cerebrum. Okay, and we'll show you some more pictures of that. The vertebral arteries, okay, that are coming up kind of closer to your neck and your vertebral column there, they provide blood to the occipital region of the cerebrum. So there is part of the cerebrum supplied by the vertebral arteries. And the inferior part, the bottom part of the temporal lobes and the brainstem region, okay, and also the cerebellum should be on there as well, okay. The vertebral arteries join to form the basilar artery before they again split off to form the posterior cerebral artery and so forth and supply those regions. We also have the dural sinuses, is where all the venous blood eventually drains into to get out of the cerebrum and even the brainstem and cerebellum, okay. So let's show you. This is, again, I like simple pictures. If you look at this one, here's the internal carotids coming up. And they go up to supply almost this entire cerebral hemispheres up here. And it divides into the middle cerebral artery, which supplies most of the outside of the cerebrum, and the anterior cerebral artery, which supplies the medial surface of the cerebrum. Here's the vertebral arteries. They come up close to the vertebral, to the vertebrae there, and they come up on both sides. They join to form the basilar artery, and the basilar artery then eventually ends as the posterior cerebral artery. Before it gives off some blood supply to the brainstem and also to the cerebellum from these basilar artery as well. But that basically supplies the whole lower part of the nervous system. You know, that underneath surface of the cerebrum and the occipital lobe, lower part of the occipital lobe, but then also the cerebellum and brainstem. So this is sometimes referred to, the internal carotids are sometimes referred to as more the anterior circulation. You know, this is more, in general, it's more anterior in nature. This is more anterior, and this is more posterior circulation, okay? Here is looking at, looking at the blood supply, looking face on at somebody here, and let's just point out a few major structures here. I'm going to go with a lighter color on this. This is our internal carotids on both sides. Internal carotids come up and they divide into this is the middle cerebral artery circulation, and they divide into the anterior cerebral artery, which, you know, forms and supplies that medial surface of the cerebrum. So that's the internal carotid coming up and supplying most of the cerebral hemisphere via the anterior and middle cerebral arteries. The most common place to have a stroke, most common type of stroke is some type of a blockage, okay? But it's very common to have that blockage in either one of the internal carotids or one of the middle cerebral artery areas is the most common area. And also if you have like a emboli, something like a clot coming from a distant location, if it finds its way up into your cerebrum, it tends to go through the internal carotids. There's a much straighter shot into the middle cerebral artery. It has to make a harder left-hand turn or whatever to get up to the anterior cerebral artery. And so it's much more common to have a middle cerebral artery stroke is the most common type. That distribution of the middle cerebral artery is the most common type of stroke you'll see, whether that's from a blockage or whether it's from, you know, or whether it's from a clot that's being released, like, you know, or a blockage that's developed slowly over time, like atherosclerosis, or even a fragment of something or an emboli from your heart going up into that tends to want to go to that middle cerebral artery. This is about, I'm about ready to throw my computer. I was this weekend, I went down mountain biking with a group of buddies down to North Carolina, and you have these, you spend hundreds of dollars on these fancy Garmins, and you got it synced up to your phone, and you have all these maps in it and everything. And the guy he was kind of like in charge of coming up with the routes and all that. His Garmin was just giving him fits, you know. I mean, you spend all this money, you spend all this time loading it all in, and you think it's going to work. We got lost so many dang times. And he at one point, I didn't know. I looked down at his screen. It was just cracked. He'd taken it and just thrown it on the ground and kicked it. And it was like a $300 piece of equipment, and it's like all cracked up. And I'm like, now what are we going to do? Now we have no way to get back. Anyway, that's what I feel like doing with this computer right now. I thought I'd figure this out. All right. So that's the internal carotids. Now let's look at the vertebral arteries. Here's the vertebral arteries coming up, each side of the vertebral column there. They come together to join the basilar artery. That basilar artery then is going to go and it's going to dive straight back. These little guys are diving straight back as the posterior cerebral arteries, PCAs, posterior cerebral arteries, that go to serve the underneath surface of your cerebrum down there. But then there's branches here. These branches are coming off here. They go to supply the brainstem, and there's a few branches that also go to supply the cerebellum. Okay? What you can also appreciate from this front end shot here is that the internal carotid circulation here and the vertebral artery circulation here, so this is the internal carotid circulation going up to form here, they are connected. They connect through these little communicating branches. These are called the posterior communicating branches back here, and there's one anterior communicating branch here. And so let's erase some of this stuff. So there's a little communicating branch. It communicates between this posterior cerebral artery and this internal carotid that's getting ready to divide into your middle and anterior cerebral arteries. Then there's a little communicating artery, really small, right between the two anterior cerebral arteries up here. So what this does, it forms a continuous connection between those different circulations called the circle of Willis. Okay? So that's the circle of Willis, if you hear that. It's that, they call it an anastomotic ring. It's an anastomosis of these different blood supplies that forms this ring. But what it allows for is, let's say, for example, and I've seen this many a times, a patient comes in and they've got a 100% blockage of their left internal carotid artery. And you're like, holy hell, shouldn't you be dead or shouldn't you be paralyzed? You know, because you basically have damaged the circulation going to that left side of their brain. However, if that blockage happened over a long period of time, like it was a slowly developing atherosclerosis, and they happen to have a really good circle of Willis that was adaptable, what happens is you can actually shift, your blood can still get around here and get over to this to help supply this part of your brain through this alternate route. It's not ideal, but if it does it slowly over time, you can get some dilation of those communicating arteries to allow enough shift of blood to maintain enough circulation. You wouldn't see a drop-off in function. However, if a person has more of a rapid onset, like a clot that happens quickly, and you haven't had time for that shift to occur, usually you still get a massive stroke. You didn't have time to shift the blood flow through that circle of Willis. Okay, but it is crazy seeing some people walk around with 100% blockage. The problem is then they start getting blockage on the other side, and you get 100% on this side and 75% on this side. Eventually, something's got to give. This is just this is that circle of Willis again right here, which I've just showed you already. Okay. Here's the question that's going to answer your question about the different types of hematomas and so forth. So, well, maybe not totally, but we'll explain it. So there's different types of damage to the blood supply and bleeds that you should just be aware of. So here's called on the left side is an epidural hematoma. Epidural, here's the dura. Right there. It's on the outside, epi. It's on the outside of the dura. It's between the skull and the dura. This tends to be a bleed that is arterial in nature. You have a few big arteries that are between your dura and your skull. The biggest one is the temporal artery, right on the side of your and that temporal bone gets a little weak in a place, and if you get whacked right on the right spot, you crack the bone and you can rip that artery. And you get this epidural hematoma. It's high pressure because it's an artery, and it bleeds very rapidly. And this is the kind of thing where somebody they hit their head, whatever, skiing or whatever, or playing football, baseball. They think they're okay for a brief moment, and they look good. Next thing you know, they just start losing consciousness. They're vomiting, and then boom, they're dead. You couldn't even get them to the hospital fast enough, and they're gone. This happened to Liam Neeson's wife, Natasha Richardson. She was skiing and it wasn't even skiing at the time. She just tripped and fell over with her skis, hit her head, and was dead in an hour, because you don't recognize, you think, oh, it's a minor thing, and then once they lose consciousness, so you got to be really close to a place, because what they got to do is basically drill a hole in your head and evacuate it immediately to get that to stop. That's an epidural. A subdural hematoma is a bleed that is between the dura and the arachnoid. Okay, so it's subdural hematoma is between the dura and the arachnoid. Those bleeds tend to be venous in nature. What happens, the most common reason you get a subdural hematoma is because you're old and your brain is shriveled. And as your brain starts to shrivel, there's actually little veins that your venous drainage goes up here, and this is the superior sagittal sinus. This is where your blood goes, venous blood. You have veins that go up and dump into this. What happens, those veins start getting stretched and they get tugged on. And if you even do something like shake your head a little bit too hard, an older adult who has a little tumble, hits their head even lightly or shakes their head, it doesn't even have to be a fall. It can just be that just walking, it'll tug on those veins, and some of those veins will tear. And those veins will start to bleed into that subdural space. And it's a low pressure. Veins are low pressure systems, so it's very subtle, and they may not even have symptoms for a while. And eventually, though, if they build up enough blood in there, then they might have symptoms, starts creating enough pressure. But they'll do like MRIs, and they'll say patient has, appears to have long-standing or old subdural hematoma that may or may not be causing symptoms, but they can cause symptoms. It's something to be aware of. Then there's a subarachnoid bleed. So the subarachnoid is a bleed that's between the arachnoid and the pia, which you don't see up here. Those are also bad because those are typically arteries that are bleeding, and they're high pressure too. So subarachnoid bleed is also not good. Okay, subarachnoid bleeds tend to be more with people with hypertension. Okay, epidural hematoma is going to be trauma typically. Subdural hematomas tend to be non-traumatic, older adults who just get stretched out veins that get pulled on and ripped. A subarachnoid hemorrhage between the arachnoid and the pia, those tend to be like hypertensive events, or an aneurysm. An aneurysm blows in that subarachnoid space, or they have a hypertensive blowout of one of those arteries. So epidural hematomas are really bad. Subarachnoid bleeds are really bad. Subdural hematomas usually aren't as bad. They can be, but not usually. Then there's one other thing called intracerebral hemorrhage. So this is a hemorrhage of deep penetrating vessels that are deeper inside the actual parenchyma, the substance of the brain, not the surface. This is down deep inside the brain, is an intracerebral hemorrhage. And I actually have one of the specimens up here. We'll let you pass it around. It has an intracerebral hemorrhage. Okay, and you'll see where it caused some damage to the tissue down, especially actually caused some damage to the basal ganglia within there, and caused some swelling in the brain. You'll see that's kind of bulged out on one side. And actually, this person, they had done an evacuation. They'd taken off part of their skull to let the brain swell and try to prevent it being further damage. And actually, they'd had the person in ICU so long, and you guys may have seen this specimen if you went down and looked at it. They'd tuck the skull down in the person's peritoneal cavity to preserve the skull. But I've got that brain cross-section, a section through it you can see. That's the blood supply, okay? Just give you a quick picture of the blood supply. Now the ventricles. This is we're support systems here. The ventricular system is these system of spaces within the central nervous system that contains cerebrospinal fluid. And the primary function is they regulate the extracellular fluid, and they do provide some protection of the central nervous system. So there's these spaces inside, but then the cerebrospinal fluid also gets to the outside and flows in that subarachnoid space. And that provides some like neutral buoyancy to the nervous system and a little bit of protection, and also, buffers some of your metabolites and so forth, and those kind of things. So it has some other functions as well. The way there's this thing called choroid plexus that makes your cerebrospinal fluid. It's like these little capillary type things. They're like these little like finger-like structures. What do they look like almost? We might be able to see some in some of these specimens. It's very granular in nature, but it's just basically the choroid plexus filters blood. It's almost like plasma. That CSF, cerebrospinal fluid, is almost like plasma. Okay, it's just a very clear fluid with very little in it, but just the non-blood components. You have the ventricles. you have two lateral ventricles, which are your ventricles 1 and 2, but they don't number them 1 and 2. They just have two lateral ventricles, but then you have a third and a fourth ventricle. You have a cerebral aqueduct that connects the third and fourth ventricle. And then you have dural sinuses, where CSF drains also into the dural sinuses. So your venous blood plus your CSF is how everything gets back into your system, okay, to be recycled. So all I want you to appreciate here is where the ventricles are. You have your two lateral ventricles. That are these guys right there that I'm outlining in yellow. This is your lateral. They're kind of C-shaped because they actually kind of wrap around with your basal ganglia and your thalamus. They're kind of wrapping around and hugging kind of those on the outside of those. There's your thalamus and your basal ganglia structures are all in here. Okay. Then you have this space right here, this little sliver of space right between your two thalami. That's the third ventricle. Then you have a little connection between the third ventricle. There's a little connection right here running through the midbrain. That's the cerebral aqueduct. And then it goes into the fourth ventricle. And remember, it's kind of rhomboid in nature, because remember there's that rhomboid fossa that it goes into. That's the fourth ventricle. And then the cerebral spinal fluid that's created within these goes through from the lateral ventricles to the third ventricle to the fourth ventricle, then eventually gets out of three openings. There's an opening on each side of this fourth ventricle, and there's one right at the base of this, right in the middle, right at the base of the cerebellum. Okay, it actually gets out. Here's one of the lateral. It's called the lateral aperture, and then there's a median aperture that's right in the middle. It comes out just below the bottom of the cerebellum, and that's how cerebral spinal fluid gets from the inside of your brain to the outside into that subarachnoid space, is through the bottom of the cerebellum. If your cerebellum gets pushed down into your foramen magnum, that hole in the bottom of your brain, it'll block that up. And that's when you get hydrocephalus. Or if you have a blockage anywhere within the connections there. So if you have a young baby that has either a problem with their cerebellum getting squeezed down through the cerebellar tonsils, getting squeezed down through the foramen magnum, or they have a malformed cerebral aqueduct, because it's so small. If it gets backed up there, all the cerebral spinal fluid you're producing here gets backed up and it starts to swell. If you're a little baby and your skull hasn't fused, you just keep swelling as the skull can stretch. But if it's your adult, you can have much worse problems because you have nowhere for your skull to go. And so at that point, you have to stick a drain, into this. You have to actually stick a tube down and a little drain that goes into the lateral ventricle, and then the tube comes down and they thread it down your skull into your neck, below your clavicle, and all the way down your peritoneal cavity to drain excessive cerebral spinal fluid that can't get out if you've got blockage down below anywhere in the system. Okay, prevent that. And they just find a spot, and they know the spots in your brain where if they insert that little tube down into there, it's not going to cause functional damage. Okay. Problem is they get clogged, they get infected. You have to pull them out, change them, and then the more times you do that, more chance of infection and risks going on with that. So this shows you the general flow. Here's the lateral ventricles going into the third ventricle. Cerebral aqueduct, that's just very vulnerable because it's so small. Into the fourth ventricle, then eventually out of those apertures, those median and lateral apertures, they get into the subarachnoid space, which has been accentuated here. It circulates around, it goes all the way down your spinal cord too, circulates around, eventually gets back up here, and there's these little things called arachnoid villi that protrude up into the superior sagittal sinus here, the dural sinus there, and that's how cerebral spinal fluid goes back in there, and it jumps in with your venous blood and gets mixed in with the venous blood and gets recirculated in your system. So that was support systems. You guys doing okay? We're just going to talk briefly about sensory and motor pathways. We're not going to get into the details here, but just the names and so forth, just the most common ones. Okay, so let's talk about somatosensation. Different ways we get our sensory information from our body back into our brain for processing. And so we have some tracts that carry this information, right? And we're going to talk about three major tracts. We're going to talk about one that's called the dorsal column / medial lemniscal system, okay? This is for carrying conscious proprioception. This is like, hey, I can tell my finger's bent about this far, or I can tell that I'm standing up, or I can tell there's how much pressure on the bottom of my feet. I'm consciously aware, or I can tell how hard somebody's pushing on my skin. That's conscious proprioceptions. That's carried in the system called the dorsal column / medial lemniscal system, okay? Pain and temperature, how hot something is, the pinprick, pain, that's carried on what's called the spinal thalamic tracts, or also sometimes referred collectively to the anterolateral system, okay? And then lastly, the spinal cerebellar tracts. That's unconscious proprioception. Think spinocerebellar. It goes from spinal cord to the cerebellum. That's unconscious proprioception. Never makes it up to our brain, okay? Those are collectively called the spinocerebellar tracts. And so my students have to learn 11 of these separate tracts. We divide there's 4 spinocerebellar tracts, there's 4 spinothalamic tracts that we talk about, but you don't have to know that. It's not actually, even for our students, sometimes not that critical. So those are the main pathways that carry information from the body. Somato, body, sensation, okay? Always look at terms and break it down. If I see a term like this, spinal cord, thalamus, oh, that's taking information to the thalamus, which if it goes to the thalamus, it's going to go to the cerebral cortex. So it's spinothalamic or spinocerebellar, sometimes if you're trying to guess on questions on test, if you just sit up and think about some of the breakdown of the terms, that can tell you where. That doesn't work all the time. The dorsal column medial lemniscal system doesn't tell you a lot, but sometimes it does tell you where it's going or starting or going to, okay? So on your left is the dorsal column medial lemniscal system. That is dorsal columns, dorsal, back part of the spinal cord. These are the dorsal columns. That's why it comes in. Here's it coming in from the leg, down lower body. It comes in. This is one of the dorsal columns. That's the fasciculus gracilis. It comes up here to the nucleus gracilis. It makes a synapse. Then the next neuron crosses over to the other side and goes up in the medial lemniscus. So it's called the medial lemniscus once you get into the brainstem. It's the dorsal columns down in the spinal cord, and it's the medial. That's why it's dorsal column medial lemniscal system. Okay, and once it gets up into the brainstem, it goes up the brainstem. It says, "Hey, I'm going to go talk to the cerebral cortex. So I got to stop by the thalamus first." Makes another connection with the thalamus, and then the final neuron goes up to the cortex in the primary somatosensory area to deliver that message, okay, about that sensory experience. This is the upper body coming into the cervical spine. It comes in. This is one of the dorsal columns. This is the fasciculus cuneatus. It goes up to the nucleus cuneatus, crosses over. And I don't expect you guys to know necessarily all those terms. I'm just mentioning the terms. I'm just giving you an appreciation how things are layered in there and how this occurs. But knowing and memorizing each of these little steps right there is not an expectation of this lecture. I'm just giving you terms that you'll hear it, and then if you read it again, it will be at least familiar to you. That's why I don't even have these labeled. I just want you to appreciate what's here without getting distracted by too much. So the main thing here is notice that with the dorsal column medial lemniscal system, the crossing over doesn't occur until this is actually in the brainstem, even though it kind of looks like a spine. This is in the upper part of the medulla. Okay, so this is the medulla. I'm going to be really patient. So that's medulla, you know it. So then this is also medulla here, medulla. But it crosses over in the medulla. Now pain and temperature, it's carried. So here's from the leg coming in. This is a lumbar segment. This would probably be L4 or L5 if it's coming from that region of the foot. Comes in, and it comes in through the dorsal root. Remember, here's the dorsal root. There's that dorsal ganglion out there. Comes into the back part of the spinal cord. It goes and actually, it may go up or down 1 or 2 levels, but it crosses over pretty much immediately in the spinal cord. So the crossing over point for the spinal thalamic tract is in the spinal cord. Then it continues up in the anterolateral region of the spinal cord and also the anterolateral region of the brainstem, hence anterolateral system. It's the spinothalamic tract, and it runs in the anterolateral part of the spinal cord and the brainstem, okay? But it crossed over, here's the upper body, it comes right in and immediately crosses over. So that's the difference here, is the crossing over points are a little different. Anterolateral spinothalamic is in the spinal cord, where it's in the brainstem up here for the medial lemniscal system. But they both cross over, go to the opposite side, okay? This is the cerebellum sitting off here. This is the this is a Golgi tendon organ, or it could be a muscle spindle, it could be a joint receptor. It's going to send a signal in, it's going to go into the spinal cord and go up the same side of the spinal cord and go into the same side of the cerebellum. So that's what's unique about spinocerebellar tracts, is they don't cross. They stay on the same side. So if I have damage to this cerebellum out here, and that would be a right cerebellar hemisphere, I'm going to have coordination problems on my right side because of that damage. However, if I have damage, you know, if I'm in like affecting the, oh God, help me. If I just had something I could point to up here, I wish I had a screen up here. If I have damage to this would be the right side of my brain. In that sensory area, I'm going to have left-sided problems with proprioception. I also could have left-sided problems with pain and temperature, right? Because those both cross over. So if the damage is, these are called contralateral pathways. The cerebellar unconscious, those are ipsilateral pathways. So damage to the cerebrum causes opposite-sided loss of sensation. Damage to the cerebellum causes the same side loss of coordination. So that's just functionally and kind of important to know. Homunculus, just to understand, there is this thing called a homunculus that our body is somatotopically laid out. This is actually the postcentral gyrus right here. We're looking now, this is a coronal section that's looking at this, and you can see how we're laid out. Here's our face, here's tongue, our arm, upper arm, a little bit back of our head, trunk, thigh, and leg, and the genitals over here on the medial surface. And so when you can damage a certain region of this and predict where you're going to have your loss of function, okay? And remember I mentioned middle cerebral artery strokes are most common, or middle cerebral artery damage. That's going to affect, oh my God, this region mostly. And you'll see people with strokes that have middle cerebral artery, they tend to have more problems with their arm and their face than they do their leg, okay? Not always, but it's very common to see that, okay? And look how, that's also the proportion of those features, or those things, that's the proportion of surface area those things are taking up that are represented there. So you see your lips, your face, your thumb, your hand take up a lot because there's a lot more sensory receptors. The density of the sensory receptors in those regions is much higher, so they take up more space. They need more processing. They take up more space in your cerebral cortex. Okay? And you'll see your leg and your trunk. You have these large surface areas but very few receptors because you don't need a lot of fine-tuned sensory information. You're not manipulating objects with your trunk. You know? You're doing that with your fingertips. Real quick now, motor pathways. We have a primary motor area, which is, there's these things called Brodmann's numbers, and it's Brodmann's area number 4. So that's this area right there. You can see the primary motor strip there. It's that's associated with what we call the precentral gyrus. And then the supplementary motor area is just in front of that and a little bit more superior. Supplementary, superior, it's right in front there. And then premotor area, a little bit more in front. Just realize the supplementary and premotor areas, those are motor planning areas. That's where, like, you've done something a long time and you've done it more than one time, you've learned it. You kind of stored those little mini programs there to tell the primary motor strip what to do, okay? Just showing you how it wraps around. Some of those wrap around in the medial surface as well. That's where your leg, the homunculus, you have a very similar homunculus to the sensory area on the surface, so you have a motor representation as well for your arm, leg, head, all that kind of stuff. We can kind of talk about motor pathways in a similar fashion as sensory. We have three kind of ways we can look at this. We have the lateral activating pathways, which are going to distal muscles. The lateral cortical spinal tract, that's the one that everybody talks about, and that's why I have it starred there. It's going to your distal muscles, okay? That's the pathway that goes and helps you can do fine, voluntary, controlled movement. There's a rubrospinal tract that complements it, especially for bringing your arm towards you for upper extremity flexion, rubrospinal tract. There's the medial activating pathways that go to mainly your proximal muscles, okay? And a lot of those actually start in your brainstem. The tecto, rubro, reticulo, vestibulospinal tracts, those are brainstem tracts. Those control muscles that are around your trunk and your proximal muscles. Automatic movements, postural movements, just to keep you upright against gravity, okay? Those are the medial activating pathways. Then we have the cortical bulbar tracts. Those are the motor tracts that go to your muscles of your face and neck, okay? Voluntary muscles, like where you're saying smile or stick your tongue out, those kind of things. Those are called cortical bulbar tracts. The brainstem is sometimes referred to as the bulb. It's bulbous in nature. That's why they call it cortical bulbar tracts. So the key ones for you is the lateral cortical spinal tract. That's the one to the distal muscles that everybody talks about. And then the cortical bulbar tracts are going to the cranial nerves, motor cranial nerves. Don't worry about that. This is really kind of a nice little, again, simple, kind of to the point little graphic display. This shows you down here, this is all of your sensory tracts. It just shows you where they come in, where they cross. So you see pain and temperature crosses immediately when it comes in the spinal cord. It goes up as a spinothalamic tract, goes to the thalamus and the cortex. Proprioception comes in on the same side, goes up the dorsal columns. It crosses over up in the brainstem, up in that midbrain or medulla area. Don't worry about light touch. It's carried on both pathways. Unconscious proprioception, this is the one going to the cerebellum. It just goes up on the same side and goes to the same side of the cerebellum. Then here's the lateral cortical spinal tract. It comes down from one side, crosses over, and then goes to the opposite side. That's all that's showing you. The little dashed line is the center of your nervous system, is the axis of your nervous system, the center point. The motor tracts cross to those pyramids. Remember the decussation of the pyramids we talked about in the front of the brainstem there? That's where those cross over. So, opposite side of the body voluntarily is controlled by the opposite side of the brain. Right? Don't worry about that. Other than this is just appreciation of all the different things that are exiting from your spinal cord and as they go through to supply your muscle, skin, and so forth. It's just a kind of a nice little simple representation of that. We've already talked about all those, but again, all I want you to appreciate here, here's the spinal cord. All this blue and red, that's all white matter. That's around that H. Remember that little H in the middle? All those are pathways. And all the red is motor, all the blue is sensory. That's how many tracts are actually running in. And that's all I want you to appreciate from that picture is that's how many tracts are squeezed into the spinal cord. And if you look at a cross-section of the spinal cord, it's about yay big. And you're like, holy hell, how the heck does all that stuff happen in there? And then see how vulnerable it is. That's why it's so protected in the spinal canal. I mean, if you damage a spinal cord, you're in a lot of trouble. You're going to really cause some problems there. So the big red one out here, this is the lateral cortical spinal tract right there. Okay. I'm just going to mention cranial nerves. You got to know them at some point, right? Everything in yellow there is cranial nerves coming out of the brainstem. Okay. There's also these two here. There's the olfactory nerve and the optic nerves that are coming out. These are coming out of the cerebrum. All the rest of these yellow nerves are coming out of the brainstem. Okay. There's all kinds of different mnemonics and different ways to memorize this stuff. But the interesting thing about cranial nerves is spinal nerves have motor, sensory, and autonomic all together. Cranial nerves can have maybe just motor, maybe just sensory, maybe motor and sensory, maybe autonomic, so they can have mixed function. But they also serve a few special functions like smell, vision, vestibular functions. So they have some special sensory functions as well. So they're a little more unique that way. So there's the names. There's the type of nerve, whether it's sensory, motor, both. and then that's primary function. Okay, and that's what you should know at least at a minimum. And then here's a mnemonic, that people use to memorize, the nerve itself, here. There's the nerves there, and then here's how you kind of memorize it by saying, on old Olympus towering tops, a Finn and German viewed some hops. The first letter of those are synonymous with the first letter of the, of the cranial nerve. And then the other mnemonic you can say is, some say marry money, but my brother says bad business, marry money. And S and M is sensory versus motor, or both. Okay, that'll give you that. So if you kind of memorize those, you can kind of know the names and what they do primarily. Okay. I learned one that I can't repeat in mixed company, but it's stuck there now, and I'm mad that somebody put that in my head, and I can't. But it works. Mnemonics work. They really do. I'm sure you've got plenty you've used in the past. Real quick, major categories of nervous system lesions. A focal lesion is like a tumor or stroke. Something where it's affecting a circumscribed, localized region within the nervous system. Multifocal lesion is something that affects multiple areas in the nervous system, like a metastasized tumor that has multiple locations where the tumor is metastatized to, or something like multiple sclerosis, where you have lesions throughout the brain, cerebrum. That would be a multifocal lesion. Diffuse lesions are things like Alzheimer's that can cause degeneration in diffuse areas, or even like bacterial meningitis that can distribute throughout the whole meninges and cause diffuse damage across big parts of your cerebrum and so forth. So those are kind of some major categories of nervous system lesions: focal, multifocal, or diffuse. The primary etiologies of nervous system lesions: trauma, just damage to the nervous system from traumatic event; bleed or a blockage, a vascular disorder, that's probably the most common; some kind of inflammatory response, MS is an inflammatory response; degenerative, Parkinson's disease is degeneration or death of cells that sometimes can be idiopathic, we don't even know why; a tumor, neoplasm, an immunologic assault on the nervous system, or some type of toxic or metabolic syndrome that can affect the nervous system. So those are really the primary. You can pretty much classify everything you see as far as nervous system lesions under one of these etiologies. Okay. Whew. Let's get some brains out here so we can do those for a few minutes here before you guys get out of here. Any questions on that? All right. So at least one brain per group, and so you guys can kind of have to gather around or move your little things out of the middle of the table possibly. So send a representative up from your group. You don't have to hold on to the brain. You guys got gloves here. If you want to hold on to the brain, you can. You can grab the brain. Just put some gloves on if you want. I never used gloves, and when I did anatomy, a guy was old school. He thought you needed to not wear gloves so you could really feel the texture of the tissue and all that stuff. And yeah, so we were doing a lot of scrubbing underneath our nails every day after class. So grab one of the ones in blue here, or I can start passing some out, but if you want to come up and grab one. No, that's fine. Yeah, these are pretty. That's pretty clean. Yeah, I kept. So yeah, you can just kind of keep the brain over that. I mean, if you drip a little bit on your table, it's probably cleaner than your cell phone. So. We'll do something real quick here. There we go. All right, so let's go through these structures that are listed here. The first is a cerebral longitudinal fissure, which you can't see because you need to have the other half of the brain there to see it. So that's what was cut down to to do this hemisection. So we do not have a cerebral longitudinal fissure. That's the thing that divides the two hemispheres right down the middle here. The next one you're going to try to find is the lateral sylvian fissure, which should be easy. That is, let me get this to yellow. That's this guy right here, this lateral sylvian fissure that divides between the frontal and parietal and temporal lobes, okay? And if you want to, stick your fingers in there and try to pull it a little bit apart, you know, and you'd see, if you can see the insular lobe or a little glimpse of it in there, there might be some blood vessels in there. One of the blood vessels coming out of that is the middle cerebral artery. It actually comes out of that lateral sylvian fissure to go to the outside of your brain. So sometimes those blood vessels get in the way of seeing that insular lobe a little bit. All right, so you got your lateral sylvian fissure. Raise your hand if you don't. The next one is a little harder to find sometimes. It's the central sulcus. So it's this sulcus that is called the central sulcus. It's usually right about here. The way you kind of know if it's the central sulcus is you follow it up to the crest of the cerebrum and follow it over the top, and if it just dives slightly backwards, I'm going to show you on the next slide. If it dives like this, slightly posteriorly, then it's probably the central sulcus. So if you go back over here, the central sulcus will come up here, and it'll dive slightly backwards like this on the other side. And that's kind of how you confirm it's a central sulcus. So make sure you get that. If you have trouble finding the central sulcus, let me know. I can come confirm it for you. Because then everything else, once we got the lateral sylvian fissure in that, everything else is pretty easy. Everybody feel like they got their central sulcus? So now that you have your central sulcus, you're going to go one sulcus in front of it. So this sulcus, one sulcus in front of it. Sometimes you have to connect the dots. Sometimes a sulcus is interrupted by a little gyri or so, so you have to jump over a gyri. But this is the precentral sulcus right here. So jump one sulcus forward to define the precentral sulcus. And now you've actually defined the borders of, this is the primary motor strip right there. That's that Brodmann's area number 4. That's the primary motor strip. That's where everything starts to send the signals, especially to the muscles that you have voluntary control over. And so the face is down. If I can, so your face is right down in here. Your hand is kind of in here, and your arm is here, and your leg is all the way on the other side of the brain. It's like it drops off on the other side, that medial surface. Okay, so that's the primary motor strip in between that central and precentral sulcus. And then you're going to jump one sulcus back, and this brain on this picture doesn't have a very good one. It's kind of like right in here is the postcentral sulcus. But most people's is a little more straight and a little more not quite that far back. But that postcentral sulcus, once you've found that, now you've defined the borders of the primary sensory strip. Okay, that's where the sensation goes to represent those regions of your body from the opposite side. So this is a left hemisphere, so that's getting all the information from the right body. And again, that homunculus thing looks almost identical to the motor one, where face is down towards the lower part, towards that lateral sylvian, then hand, upper arm, trunk, and leg going over the medial surface. Okay, that's the primary sensory strip. So we've got those defined. Let's look for one other thing. There's a thing called a preoccipital notch. It's a little notch right here. So here's your cerebellum down here. So it's about 3/4 back on the cerebellum, just above the cerebellum. It's this little notch. And some brains have a more appreciable one than others, but that's the preoccipital notch. And that's going to help define where our occipital lobe begins. And so if we have if we have our lateral sylvian fissure, our central sulcus, and our preoccipital notch, what you can do is you can kind of take an imaginary line up from the preoccipital notch and kind of go straight up from there, and you actually connect it to this area here, which is the parietal occipital sulcus. You connect it on the other side, and then you just extend this lateral sylvian fissure out, and now we've got frontal lobe, parietal lobe, temporal lobe, and occipital lobe back here. Okay, so that's kind of divides that up. If you, that Wernicke's area I told you about, that's kind of right back in here. This is that lateral sylvian fissure. It's kind of wrapping around this lateral sylvian fissure. That's that area. If you have a left hemisphere, which some of you have right hemispheres, if it's a left hemisphere, that's where that Wernicke's area is. That's that area for understanding language, okay? If it's a right hemisphere, that area is for understanding nonverbal language, okay, body language and things like that. That Broca's area is right around this thing right here, this area. This little area right here is called the operculum, this area right in here, but Broca's area is right here, and that's the speech center. That's for planning speech, expressing yourself, is that Broca's area is right in there. If you have a big middle cerebral artery stroke that takes out both those areas, that's called global aphasia. That's where you can't understand things and you can't speak either because you've damaged both the expressive and the receptive parts of speech. It's called global aphasia. And that is somewhat common to have a middle cerebral artery stroke that can kind of knock both of those out. Superior temporal gyrus. There's a gyrus right here. This is the temporal lobe. This region right here is a superior temporal gyrus. Right here is a region that I'm darkening in. That's the primary auditory area. When you first hear a sound, it goes to that primary auditory area, and it defines it as a certain type of sound. It like goes, oh, this is a sound, and then it sends that sound up to Wernicke's area to make meaning of it. You know, it categorizes the sound and then sends it up to the Wernicke's area to say, oh, the meaning of this sound is, oh, this is a language that's telling me this, or this is a bell that's ringing, or this is, you know, so that's where, but it goes to that little spot first, the primary auditory region. Okay. Now, we're going to flip the brain over to the medial surface. All kinds of fun things on the medial surface here. The first thing we're going to show you there, and it's very easy, is the corpus callosum. That's this big structure here that joins the two hemispheres together, runs the whole length of that, so it really joins mostly the frontal and parietal lobes together. It's got different parts. It's got a little rostrum up here, the little beak of it. It's got a bend, or called a genu. The body is the middle part, and then the splenium is back here. That little round part at the back of it is called a splenium. Not that you guys have to know that. Just know that's a corpus callosum. Let's highlight the diencephalon now. The diencephalon is this whole region right here. But we can further subdivide it into this region right here is more of the thalamus. And then below is the hypothalamus. Some of you might have a little bump right here. Sometimes that's the intrathalamic adhesion. There's a little connecting stalk between the two thalami that are sitting right close to each other on each side. There's a little connecting stalk between the thalamus. But depending on where I got the cut, you may not see that. I might have cut that off, okay? This is actually also your third ventricle. When you take and you place the two thalami close together, there's a little sliver of space in there, in that diencephalon region that forms the lateral walls of the third ventricle. There's a little space in there that's where the third ventricle is. Okay. Don't worry about the anterior commissure. Let's go to parietal occipital sulcus. Back here, there's a really prominent sulcus right here between the parietal lobe and the occipital lobe. Parietal occipital sulcus. Okay, that's a pretty easy one to find. Staying back now in the occipital lobe region, we're going to define, this is called the calcarine sulcus right here. There's a sulcus right here called the calcarine sulcus. It divides the the occipital lobe into a cuneus up here and a lingual gyrus down here, lingual gyrus. The only thing I want you to know about that is that your vision, when it gets back to your cerebrum, your vision goes to basically the little lips or edges of this sulcus on both sides. This is where your primary vision is. And your vision is upside down, and it's the opposite side. So, like the vision that goes, this is a right hemisphere. So this area right here that goes to this would be seeing the lower part of your left vision. Even because so this is on the upper part of your right hemisphere. That information getting to that is coming from the lower half of your left visual field. So don't worry about it. It's kind of weird how that happens. Why it does that? It's the opposite side, and it's typically a little just different than what you would expect it. So these are the two. This is the areas where primary vision comes to. Okay, when it first makes it to the cerebral cortex. This right here. There's a sulcus right here, right above the corpus callosum, called the callosal sulcus. And there's another sulcus that's right here that I'm following called the cingulate sulcus. Okay, and actually that callosal sulcus extends there, and then what we defined here is the cingulate gyrus. It's kind of C-shaped, cingulate, C-shaped. So it's the cingulate sulcus. That has to do with limbic system function, controlling emotions and so forth, regulation of emotions and so forth is the limbic system there. Down in here, some of your brains may not have this. There's a white structure right there, and there should be a little tag end of an optic nerve coming off of it. This is the optic chiasm. This is where some of your visual information crosses over to the opposite side. And that's how you get that opposite side of visual information from one field of vision going to the opposite side of the brain, is that's happening in that optic chiasm. So the optic chiasm right here, and there's a little tag end of an optic nerve coming off there. Pituitary is actually, so there's a stalk that comes off the bottom of the hypothalamus right here, and the pituitary gland is right here. However, you can't get it out because it's stuck in the skull. It's in this little divot called the sella turcica. It's got a little membrane over it, and so as you pull the brain out, you always rip it off. Yeah, so you can't really, but that's where it would be, right there. And that little stalk, sometimes some of you might have the little stalk there. It's called the infundibulum. It's a little tiny stalk that comes off there. Most of it gets ripped off, though. Right at the bottom of the hypothalamus there. Some of you will see the pineal body or the epithalamus back here. This is the pineal gland back here. This secretes melatonin. This is for regulation of sleep and some of yours, if you poke into it, might be like kind of hard and calcifies. As somebody ages, they get more calcified, and that's why as you get older, you sometimes don't need as much sleep because you don't secrete as much melatonin because they start to have that loss of function of that pineal body or pineal gland. Some of you will see this. There's a little white structure right here. So here's the optic chiasm. You come straight up. This is called the anterior white commissure. That's a little axons that go from one temporal lobe to the other, cross over. That's the anterior white commissure. There's a little bump right here. This is the mammillary body sticking down. This is actually, now we're defining kind of, this is the midbrain right here. Let me erase some of that stuff. It's starting to get a little messy. So let's just kind of, this is the midbrain right here, which has the mammillary body. This is the interpeduncular fossa right here. And then, see this region back here? That's that superior and inferior colliculus. Think sagittal cut through the superior and inferior colliculus. And so this is the superior colliculus right here, and this is the inferior colliculus. Normally we were looking at them from the back like this, okay? and then this is all midbrain. The superior and inferior colliculus in this view is sometimes referred to as the tectum of the midbrain. It forms like the roof over the midbrain. A tectum is a roof. This is the basilar pons, that big round front part is called the basilar pons. And then down here, this is medulla, down here. Here's your cerebellum with the anterior lobe and then this big posterior lobe of your cerebellum. Now most of you, some of you, this brain still has a membrane right here. There's a membrane covering over this just below the corpus callosum. If you have that, you can kind of push it out of the way with your probe or something, or stick your finger in there. Most of you don't, but if you look right in there, that is one of the lateral ventricles. That's the lateral ventricle where cerebrospinal fluid is at. And you may see this little granular substance like right here in the floor of it. That's choroid plexus. That sits often right there. And there's often some choroid plexus down in the fourth ventricle too a little bit. But you may see some of this little granular substance. So the cerebrospinal fluid comes out of this lateral ventricle, and there's a little hole right here. There's another white structure here called the fornix, but it comes down through here, so it goes out through this. This is called the interventricular foramen. Goes out into the third ventricle right here, goes down through the cerebral aqueducts. This is the cerebral aqueduct right in there, just in that midbrain. That's that vulnerable area that's very tight for that cerebrospinal fluid to get through. Then it goes down to the fourth ventricle right there between the pons and the cerebellum. That's the fourth ventricle down in here. And then eventually it goes out from below the cerebellum in the middle, down in the middle through the median aperture, and then out the two lateral apertures to get into that subarachnoid space to start circulating around the cerebrum. So, and then I'll pass this brain around. You take so this brain here that I'm going to show you is cut in a horizontal section. The stroke is on the left side, and you can see where the brain's like, bulged out to that side. And you can see this damage, kind of this empty area near the basal ganglia structures that's really kind of cavitated and divoted out a little bit, where there was actually a bleed in that area. And you can actually see in some of the other structures cuts where there's some of the blood and so forth, but not this one. But anyway, you can see where that damage was. I'll let you guys kind of pass it quickly, and you can just take it to the next group once you've kind of had a chance to appreciate that. And then I'm going to start. This other one is a coronal section. And for this one, you'll want to just kind of appreciate where you'll see the thalamus on each side, and you'll see the basal ganglia structures a little more lateral on each side. And you'll see the lateral ventricles and so forth. So I just wanted to give you appreciation. And you'll see the third ventricle in the middle there a little bit. You actually see a little intrathalamic adhesion is in there that's in the middle of that third ventricle. So you guys can take a peek at that. I'm just going to walk around. If there's anything you didn't pick up or you have a question about, I can kind of like point things out. Let me pause my recording right now before I do that. But